Welcome to another video for biology. In this video, we're going to be discussing sex cell formation, or meiosis. Okay, meiosis is a little bit different than mitosis. Okay, and as you can see here, here's some of the objectives that we're going to be discussing, some of the questions that you need to make sure you understand at the end of this video, or questions that I need to make sure I explain by the end of this video. So let's just go over these three main objectives. Number one, how do sex cells form and what is a sex cell? What are the stages of meiosis and how does meiosis provide genetic variation? So let me go ahead and start off by answering the first one. How do sex cells form? Well, they form by a process called meiosis. Now, what is a sex cell? So if we look at a human being is made out of, we've discussed this in class before, 46 chromosomes. Okay, 23 chromosomes, two pairs of each, making 46. Okay, so each human being gets their chromosomes half from mom and half from dad. Half of the genetic information that you've gotten from dad, and the other half you've gotten from mom. Okay, so this particular video discusses how a cell, now we've discussed mitosis. Mitosis splits into two identical cells, but meiosis splits into four cells, each having half of the amount of genetic information. And they're not exactly the same either. So a sex cell is a cell that has half of the genetic information that you have. Okay, and you form these every day. Women form their sex cells in their ovaries. Okay, and males form their sex cells in their testes. Okay, and then when you have these two meet, you have fertilization, a zygote, and then you begin to have the formation of a human being. All right, well, let's begin. Interphase. If you recall from mitosis, interphase is the process in which your organelles are divided and your DNA is, I'm sorry, not divided, but they're replicated. And your DNA is also replicated. And it's replicated and it condenses into the form of chromosomes. So those chromosomes each have all the genetic information that is made, that makes you. And at the same time in prophase I, so prophase I is signaled by the chromosome formation and your spindle fiber formation from the centrioles. Okay, you also have a process called synapse where you'll notice that the chromosomes, in fact, let me back up a little bit. You'll notice as these chromosomes come together, you see those little dots, dotted lines. This is synapse. This is where those chromosomes come together, homologous chromosomes. In other words, these are chromosomes each that have the same genetic information. They're combined by synapse. And what ends up happening, if you look at the chromosomes, at the point of connection, you have certain segments of DNA that are switched or they're crossed over. Okay? Now, if you think about this for a second, now these two, or this process crossing over, gives genetic variation or helps with genetic variation because now you have chromosomes that have, at certain areas in their code, have switched. So that allows for the chromosomes to actually be different from one another. All right, so as prophase continues, you'll notice that the nuclear membrane will break apart and the spindle fibers will continue to form. Now these spindle fibers will attach at the centromeres of each chromosome and they will line them up. Okay, they will line them up at the equator of the cell. Now notice here that in metaphase I, you have your homologous chromosomes, each pair of chromosomes lined up as pairs, as opposed to metaphase from mitosis where there's a single line in the middle. Okay, so mitosis, you have the chromosomes lined up at the middle, and then in anaphase, you have the process of these chromosomes being pulled apart at the equator or from the equator to the poles. And then telophase stage, you have the reformation of the nucleus. Okay, and you'll notice that the cell membrane is also forming as well, making two cells. Okay, so now you have a process where you actually, before you go from telophase I to prophase II, understand that from telophase I to prophase II, and I don't have this stage in there, but you have another phase of interphase. Okay, interphase, unlike the previous stage of interphase, you do not have a replication of DNA. Okay, so in this second interphase, there is no DNA is not copied again. Okay, so from telophase I, we move over to prophase II, Keep in mind that meiosis is a process of a two-stage cell division. Mitosis was at one stage of cell division where it copied into an identical cell. But with meiosis, you have one stage which we've just been through, which was prophase I all the way to telophase I, and now we're entering the second phase of cell division. Okay, prophase II, and then much like with prophase I, you have the same thing. You have the spindle fibers that are forming. Okay, the centrioles going to the poles, and then with metaphase II, you end up having, okay, and they meet here at the centriole, the center part of each chromosome, sorry. So the spindle fibers will now align the chromosomes along the equator, and you notice again here, unlike metaphase I, metaphase II, they line up the chromosomes in a single line as opposed to paired up. Okay, so anaphase II, these cells are separated. Okay, now something interesting about the process of these and how these cells separate in the stages of anaphase, and this goes for anaphase I and anaphase in mitosis, is that this process, it's really actually pretty cool, and I'll post a small little snippet of an animation showing how these chromosomes are actually pulled apart.
We're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have two strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes. We have no idea really, as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red, and it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way.
So now we move on to telophase II, where the chromosomes, and understand that you no longer have the cell that you had before. You do not have a complete set of genetic data in each of these cells. Okay, each of these cells now has half the amount of data that it had when it first started off. And you'll notice if you take a look at these chromosomes, these sister chromosomes, that you'll notice the chromosomes that had the crossover in back in Prophase I, that they are not the same as the ones that are across the way. So you have each of these cells has half the amount of genetic information, and they are very different from the original genetic information that started off. So this is a process that helps us, going back to the objectives, this helps us in giving more genetic variation to the human being. So you don't have a baby that looks exactly like mom or exactly like dad, but you might have a baby that looks part mom, part dad, and grandpa or grandma or great grandpa. So this process allows us for more genetic variation. Okay, so now you'll notice in telophase that the centrioles will degenerate, and so will the spindle fibers. Okay, so again, each cell has half the amount of genetic information. Your nucleus will begin to reform, and you'll notice that instead of having 2 cells now, I am forming the cell walls for 4 cells. And each of these cells is called a haploid. These are gametes. These are sex cells. This cell has half the genetic information that a human being needs. And again, I form 4. Okay, so again, they're haploid cells, and in order to make a human being, I need the other half of the genetic information, which either the mom or the dad will contribute. And then once they fertilize, that will make a full new human being. So again, this has been the study. Okay, we've been looking at meiosis. This is sex cell formation. This is how cells divide and genetic information on one cell, and the other human being provides the other half of that genetic information. I hope this video has helped. Good luck in your studying.